Good morning, everyone. I hope I'm audible.
Yes, sir.
Yes, good. So in today's class, we will discuss about a few things, including we'll start with screening, but we'll also, it is a kind of mixture of several techniques. Okay, so we will learn some fundamentals about screening, and then we will also learn some things about um, one second. Yeah, we'll also learn some things about the libraries where we have left off something like race and other things. Okay, so but they're all but they are still interconnected. So the first thing is, when we say screening, what is we kind of, it's better to have a kind of definition for it. Screening is something to you have. Say, for example, there is a mixture of several things around, and just it's an abstract thing. Say you have A, B, C, D, and so on. I want to find one particular aspect out of it, one entity. Here I have A, B, C, D, F, G, H entities all as a mixture. It's like trying to get get what I need. Say I want to know if there is. I want to find out which of these is D. You're trying to screen through all the things that are available and try to find out what you need. As in the case here, I need D, entity D. Some years ago, it's like it is like finding the needle in the stack or haystack or whatever they say. Sorry, something like this. You have so many and you have to find out, and that's actually a very difficult one. There is a counter to this. If you want to do the straw, probably finding the needle in the straw, I think that's what it is probably. So what you do is to burn down the straw so that the needle is the only one that doesn't get burnt, and then you take it and use it. Just I heard it in some movie or some documentary. But screening is about finding what you need from a large group of things. Here I have put only a few of them. If these entities are thousands or even hundreds, and how to pick one. If I have to search one after one and pick this out and see whether it is or not, and then pick the next one and see whether it is what I need or not, it's going to take a lot of time. In one go, I want to find out. And these are all those techniques involved, especially in the molecular ones. You are trying to see that is screening basically. First one is we will try to, we started constructing a recombinant DNA, wherein we had the vector and we using many other techniques, and then finally ligation, we made a recombinant vector. But you also may have vector religation. We have techniques for reducing the vector religation and so on. Even then, I want to find out if the vector, the sample that I have, which may constitute both a recombinant. This is a recombinant, and the other one is a vector religation. How would I find out which one is of my interest or not? Then you can use restriction mapping. Restriction. I think we haven't given you the restriction mapping problems. Please remind me. I will give that to everyone. Restriction mapping is using restriction endonucleases. This is the marker or ladder. And here is empty vector that you already have, which you are planning to use it as a vector in the situation that you're doing, in the context. And here is a clone. And when you say, I will make a setup so that you will understand. Here is a vector, BAM H1. I have digested the fragment so that it also has BAM-H1 overhangs. So both were actually digested with BAM-H1, then mixed, and then ligated. So there are two possibilities. In one case, you may have vector religation, and in other cases, you may have vector and the fragment along with the fragment here. Assume that's the fragment. Now, if I have several, I have multiple of those. So vector, I am giving it to be 3 kb. The one that I have chosen here, it's assumed that it is 3 kb. Fragment is around 1 kb. Okay. I'm going to draw a gel diagram, and from that you can figure out very easily which one is and not. Say, sorry, 1 second. Okay. So if I have put in vector alone here, and it comes 3 kb fragment will be there, I will give you three different clones, clone 1, clone 2, and clone 3. So clone 1 is giving, I have digested these clones that you have here. I have digested with BAM-H1. I could have also used other restriction endonucleases also based on the context, what restriction enzymes are present. And then we have to, I got something like this upon, if I have, oh, sorry, I made a mistake. I assume don't count that. What can we say now? Just forget about this. We have 3 lanes. In lane 1, I have vector alone, and say the marker is something like 1 kb, 2 kb, 3, 4, 5, and so on. Empty vector that I know of allowed, and it gave a band at 3 kb. That fits with the experimental setup that I have taken here. Clone 1, I have digested it, and I found only one band that is corresponding to 3 kb, which means it doesn't have any other DNA, which would mean that it is a vector-vector religation. Similarly, I have done for sample 2 or clone 2, and I found 2 bands. One is of vector, and there is another one, 1 kb fragment, that is of fragment. So out of these out of these 2 clones, for simplicity, I'm just taking 2. Out of these 2 clones, 2 is recombinant and 1 is vector-vector religation. Okay, so that is one of the simplest way. But one can also do using probes, right? Like we have done in southern blotting. We blot it onto nitrocellulose membrane, and then we use a probe. We will discuss a little bit about a probe as well. And then when we do blotting, we will find out that it gives a signal. Whichever clone gives signal must be the recombinant. Okay, these are simplest techniques that we know of that people can do. They do routinely for confirmation purpose. These tests are usually done. Now we will, this is the one that I was describing a little ahead. Here, it's not about one clone or one ligation. We are doing for a whole library, right? What is a gene library? If we had a genome consisting of multiple genes in it, and by definition, I will repeat again what a gene library is. Gene library is a collection of clones, a collection of recombinant clones, which have all the genes of the test organism represented at least once. That means we must be having a clone like this, which had gene A or fragment A. Here should be B, C, and so on. Like that, D, E, F also. Okay. So that is a genomic library. Now, if you have constructed genomic library, what do you do with that? You need to, we are constructing it for a purpose. Usually the purpose is about trying to find the gene of interest, which may give a product. The presumption is that we actually don't know the order of genes. We don't know the whole the function of genes. We don't know the sequence. So typically the goals will be to know the sequence of the genome, to know the, I mean, the same thing, the order of genes. How are the genes? Which gene is after which gene, and so on. You'd also want to characterize the genes. So typically these are some of the experiments, but reasons why we create libraries. Okay, but there are also many others. You want to create, you know that you have that say for example there is a fungi, and you know that it produces say amylase. Sorry, amylase. And if you want to do that, how can you make how can you only know the sequence of it, sorry, you only know the property of it by, I think you people are doing experiments as well in many laboratory experiments. You know that there is, you can isolate a fungi that is producing amylase, but I don't know anything about its gene. All we know is that the organism produces it. How do I get the gene for it? Right? How can I get the gene? How do I know? If I want to know the amino acid sequence of it, I could produce a large amount of amylase, then go for protein sequencing. Right? You can do it protein sequencing using normal degradation methods, Sanger's method. Sanger also had two Nobel Prizes, I think, one for DNA sequencing and the other one for protein sequencing. But there are also now easier and better methods using mass spectrometry. Okay, that's a very good area of research as well. But otherwise, what we can also do is to know the gene, and to be able to find the gene is the point. And in that case, the approach will be typically one would take an approach like this. You construct a genomic library and see which of the clones actually produces amylase, right? If there is, say, for example, gene B is the one, this clone is the one that has the gene for amylase. Maybe upon screening, how would you screen for it, right? You would screen using a plate. You might take nutrient agar or whatever, and then you might also add amylose in the medium, so that if you are assume that this organism, whichever we chose, can produce can secrete amylase and so on, and then you can put it in onto iodine or other kinds of indicators which would bind to amylase. So if you find a region where there is clearness, if it is clear, a zone of clearance kind of thing, then that means that might be producing or somehow hydrolyzing the amylose, which means that particular clone is the one that is producing amylase. That means it must be having the gene for amylase that we have constructed through genomic library. Okay, so in here you are assuming that you took a host. Host by itself cannot produce any amylase. You should have had taken that care. Host is amylase producing, or negative for, or mutant for producing amylase. So here is a biochemical test through which you can screen a large library for the objectives that you have. Here the objective is to find the clone that has that is encoding amylase. Once you identify this, you take that out, you can do restriction diation, you can go for sequencing, and now you are characterizing the whole gene. You can also look into its promoter, if there are introns, any of those things. Okay, so there are a lot of applications that genomic libraries or any of these libraries are for. That is why we are doing these. Now, of course, as I said before, we directly go for sequencing because we don't have to construct genomic libraries. We can directly go for sequencing and we have bioinformatic tools to annotate and identify the functions of genes, at least putative functions, which now using PCR we can clone those particular genes or sequences and then characterize them. So the same in that way, we also need to know the molecular. There are also there's people still do use them, molecular clones, where if I have the construct like this is a clone. Plasmid preparation was done. This is a library which is done in BAMH1. This is to assume that you have each of them must be having a fragment, something like this, with BAMH1 sites like that. Those that are vector religation may not have the fragment, but they will just have this BAMH1. So out of these, we need to find out which of those are actually recombinants and which are not recombinants. There is no point why one should keep carrying any vector religation ones. These should be eliminated from the library. Otherwise, they're just adding to the number. So what you do is you take the clones one after one, you digest them with BAMH1, and then you load them up onto the lanes. Each of the clones will give a fragment for vector that's usually on the same place, say. But then those that are recombinant will also have additional bands like this. Say in this lane, in this gel picture, clone 5, okay, that is loaded on lane 5 here as mentioned, that does not have any fragment indicated, which means that is a part of the vector religation. So if this is a good library, why is that a good library? It is a good library because you do not have out of 6 different samples that were tested, what you have is only one of it is a vector religation, this one. So the rest are all recombinant, which would mean that is a good library. Okay. If we had that majority of them are vector religation and only 1 or 2 are these recombinant, then it is not a good library. It is all about, when you say library, we want to have it as small as possible, but libraries tend to be large in the sense that the number of clones that you would generate will be huge. That is mostly because we want to increase the probability with which each of these genes should have been represented at least once. We want more than 95% probability or 98% probability. So when we are going to do something like screening, sometimes we know, in this case, for example, we know what is gene A, that there are fragments, but we don't know what they are, right? We have to characterize. Say, for example, I know a little bit, and then I should be using probes, right? I'm now going to speak a little bit about probes so that you will understand this is applicable to southern blotting, it is applicable to northern blotting, and also for screening that we are going to look at today. That's screening the whole library, okay? So first thing is, I also had this, but I'll just speak about this one. First thing is, we have a library and a DNA. It's usually double-stranded. Now assume that this was already on the blot. We have done electrophoresis, then we have blotted it onto nitrocellulose membrane. Then we have several steps in there. First thing is, if I add a probe, say for example, I added a probe here. The probe is complementary to this, okay, complementary to the strand, the bottom strand. But then the bottom strand is already base paired with its complementary strand. So there is no place for the probe to go and bind here. So one of the steps that is included in this case is that you have a denaturation step where the two strands are separated, right? You do not add, prior to addition of probe, we have denaturation step and fixing, so that the double-stranded DNA would break up and open something like this. And then the probe is usually added. Now the probe is added because it is single-stranded denatured. The probe can now base pair. Again, looking into it a little closer, where does it bind? Where does it not bind? I will draw a sequence of the probe, right? A, G, A, C, T, A. I've taken a small probe, and I'm also going to take a DNA molecule, a little bit longer. And I'm going to write okay. Where do you think this probe will bind, and where will it not bind? There are two possibilities. Say, for example, I think I tried to write, this is one sequence, right? 1, 2, 3, 4, 5, 6. So this is absolutely complementary to this sequence. So there is another sequence also I have written somewhere here. Okay. So here, in this case, I'm talking about very small sequences. So I will write here AGA CTA. That is one place where the probe is bound. There is also a possibility that the probe can bind here, except that there is one mismatch. Here is a mismatch. The rest of the DNA is base pairing, right? This is the best sequence, best possible base pairing. That is the absolute one. All of it are matching. This is the next best one. In the sense, out of 5 bases, out of 6 bases, 5 are base pairing. There is only one mismatch. And this is always a possibility. You may have, this is highly specific, whereas this one is little bit non-specific. If your stringency conditions are not good, what may happen is here it's only one mismatch, but it may also base pair where there is more than 50% base mismatch. Then your whole analysis is going wrong. So what one should do in that case is we should know, we should be able to wash we should have high stringent. Say, for example, I am going to increase the temperature. If I'm going to increase the temperature, which one is going to fall off first? If I'm gradually increasing temperature, the non-specific bound ones are usually the ones that fall off first. Why do they fall off? Because they are bound by less number of hydrogen bonds compared to the other one, isn't it? In this case, you have more number of hydrogen bonds compared to in this case. So if stringency is about how we increase it is about temperature. It is also about salt concentration and other buffer conditions. So one should know for each of the probes and the experiments, we have to optimize what is a good temperature or what is a good stringency condition. If I increase too much, then even this probe also can fall off from this side, and that is not what we want. We want to have such that the highly specific ones are still hydrogen base pairing with their respective sites, complementary sites, but the non-specific ones may fall off. So with probe, this is something one has to pay attention when you're doing any of the blotting techniques or hybridization when you are doing it. This is to avoid non-specific interactions between probe and the test sample, but also retain the highly specific base pairing as well. So there is one more aspect here. As I told, if I didn't know, if I don't have any knowledge about this sequence, what I knew is, as I'll continue with what I have discussed before, amylase, I have found that an organism produces amylase. I don't know any idea about its genome or gene sequence, but I can find out the protein sequence using protein sequencing techniques, and or a small peptide of it. Say something like histidine, its sequence is like histidine, phenylalanine, proline, phenylalanine, and MET. Then what can I do? I can synthesize a probe based on that. How should the probe be? It should be CA, T, or C. Okay, I will just put it like that. I will try to write a little bit larger so that you will be able to understand it better. So I'm just rewriting this probe based on something, CAT, right? T or C in the third position. Then T, T or C. Then C, C, and it could be N. N means any nucleotide. Then you have T, T, T or C, and then A, T, G. If I have known the protein sequence, I have to find out the region which has more number of amino acids that are represented less number of times in the genetic code. I hope you remember the genetic code. We have 20 amino acids and about 61 codons representing 20 amino acids. Right? That means some amino acids are represented by more than one amino acids. Codons. So we should identify within the protein sequence a place where amino acid region is rich. Protein sequence is rich in amino acid residues represented by less number of codons. Say, for example, C for histidine. It is CAT or CAC, right? So when you're making a probe, you can do something like this, CAT or C. I hope you remember how we construct the gene synthesis, right? When we are doing DNA synthesis, what you do, you will add first CA, then T, DATP, then you'll get A, then you have to add either T or C. So what you'll do, you will have DTTP and DCTP. So that in some cases, T will be added, in some cases, C will be added. Right? I hope you remember when we discussed about oligonucleotide, doped oligonucleotide in mutagenesis, in cassette mutagenesis, there we use something like this. So the idea of what we are trying to say here is, if we didn't have any knowledge about sequence of the DNA, right, and yet we want to identify the clone, then the logical progress is to identify regions in the protein sequence and know the place which is represented less number of times in the genetic code, and then we should be able to synthesize a probe like this. When we use a probe like this, it may give more number of bands because it's a mixture of bands. But then we have to slowly characterize and identify each of these, and then probably we will find out which one is giving a producing amylase. Okay, these are some of the things that people in the earlier days they used it. Again, my objective is about how we think, the flexibility, and the within ourselves, how diversely can we think. That from there we come up with the best solutions. So when we know about how problems were solved in the previous days when the technology was not good, that is an important skill each of us have to improve. It's about problem solving and troubleshooting things. Research is all of it. Research is solving a problem, or and during the course of the research, one will come across a lot of problems or snags. How we solve those troubles is the point. Okay, now assume we have made the DNA probe is made, and this is something called as here we are screening a gene library by hybridization on the plate. We are not doing electrophoresis and all those things. We made a clone, and each of it, we made a gene library. Each clone is representing means it has it is made up of cells, each cell having a plasmid like that. Okay, it could be a recombinant or a non-recombinant. And from this library, from this plate, I want to find out how which of these clones is of producing amylase, for example. Okay, so what we do is we make a filter of it. We'll make a filter, and the cells from the plate will be transferred onto filter, something like this. And then there are a lot more steps that we will look into in the next slides. But the simplest way is to, once you do that, you have to actually open up the cells, and then the plasmid is there. That plasmid should be denatured. There are a lot of steps involved. Okay, and then you will add a probe. So this is a filter. This is the plate on which the library is present. We picked up these colonies such as represented here, and then we have processed it by fixing the cells, lysing open the cells, denaturing, and then hybridization with the probe. Whichever colony gives positive signal, that is the colony that has the recombinant plasmid that we are with the gene of interest. And then we take that and we go for further processing. But the objective is simple. As I said, try to define screening. You have a lot of things, and you want to find the interest, what you are interested in from a large number of entities. This is the same, okay? But this is, we don't have to memorize all this stuff, but the actual or complete process is given here. This is the plate consisting of the genomic library. One has made a replica plating and then transferred onto the nitrocellulose disc. Then we are going to, as I was tackling in the previous one, we are going to lyse bacteria using 0.5 normal NaOH. Then we have to neutralize and then add proteinase, because the nucleoid is with lot of proteins also, something like chromatin in bacteria. And then we have to wash it and then bake at 80 degrees centigrade. There are two things that will happen: that the DNA, the cells along with the DNA, they get fixed to the filter, and also that the DNA could be denatured. It's usually done overnight also sometimes. Then you're going to add P32 labeled probe. I hope you remember how to make labeled probes. We have discussed this when we were discussing some of the PGEM vector, which has, I think you have to look back into those slides or videos and try to figure that out. I have discussed about random labeling, random radio labeling. I haven't discussed end labeling yet. I will discuss end labeling when we are going to read the corresponding technique. So one will add a probe. After that, there will be a washing step and then autoradiography. And the colony where, because you are likely to have more number of plasmids, okay? So each cell, one colony is made up of several thousands of cells, okay? Thousands. And in each cell, there is maybe the copy number can vary between 5 to 10 or 13. It depends on the origin of replication of the plasmid. Assume 10. So you have a large number of plasmids uh, in there. And when you add the probe, it might bind to some. And it is radioactivity, which can give, which can amplify the signal several fold. So you will be able to identify the colony on the master plate, which may have the uh, gene or sequence of interest that you are choosing. There is another method. The method that we have discussed here is for colonies. Okay. The next method that we will see is for, it's called as okay. The method, this one is called as colony hybridization. If I didn't mention that, colony hybridization. The second method is called as plaque lifting. Okay, I hope you remember what is a plaque. You will have a background of cells, and then in some areas it is clear. Why is it clear? It is because the phage, it has lysed the cells around, so you'll have clear zones like this. That is referred to as a plaque. So this is a technique used when you are making a library in something like lambda phage, for example. So the principle remains almost the same. There is no big difference, except that now instead of cells, you have plaques. So you make the plaques, then you lift the plaques using a nitrocellulose sheet, same as the filter what you have done before, and then you add, you retain the plate. You just take the sheet, and then you're supposed to treat it first with alkali to denature DNA. There should be one. And then they neutralize the filter paper, and then bake at 80 degrees centigrades, and then hybridize with the probe, and then autoradiography, washing, and then autoradiography. It's the same thing, basically. It's not so much of a difference. In some cases, I think I wanted to mention that. I'm looking for it. Say, in some cases, how do you open the cells, lyse the cells? In some parts, people actually also use phages to open up the cells. Okay? Those techniques, so people have done that. So you have a mixture of a spray of phages, and you spray it onto the filter. And hopefully it will get in there and lyse the cells open. So I will take this last technique. It's a kind of introduction. You must have read it before. I wanted to take subtract to library and race, but I will just stop with subtract to library. The whole principle of subtract to library, or the need, arises from something like this. Say my research objective at this point is, I have cell type A and cell type B. I want to find the difference between these two. There is a genetic difference between these two. I want to find the genetic difference. I don't want to. So one of the strategies, or the long cut, maybe it's like this. You make a library of it. I make a library of it, then characterize each of the clones and see what are all the genes that is present in cell type A or organism A, and then also take a library and then try to analyze what are all the genes present in it. Then I can make a comparison between the genomes of A and B, right? Then I will be able to find out what is the difference. There is also another way of how you can do it. Is the questions also may arise. You have subtractive libraries for gene libraries. You can also have subtractive cDNA libraries. We are not going to discuss about subtractive genomic libraries. We are only going to discuss subtractive cDNA libraries. It's simpler and better probably. Genomic libraries is a little more complicated, but we don't need that at this time. So my objective now is I'm changing it. I have two different types of cells, and I want to know which genes are expressed in cell type A and which genes are expressed in cell type B. I want to find the difference alone. Okay, I don't need to know all the every other genes, because if I have two different cells from the same organism, there will be some genes are expressed in both. Some are expressed only in A. Some are those that are A specific, or sorry, A specific. I will write B specific genes. B type specific genes are expressed only in cell type B, but not in A. A type specific genes are expressed in A, but not in B. Some of them are constitutive, okay, in the sense that something like important for glycolysis or Krebs cycle, they have to be expressed in both the cell types, because all cell types depend on energy generation from glucose. And there will also be some genes that are not expressed in either of them, right? Say, for example, cell type C specific cell type. So only cell type C will, in those cells, these genes will express. These are not, this is A and one is B type. So they don't have to be expressed here. So we all the cell types that we have in our body will have differences. Those are all transcriptomic differences. So some expressed in both, some expressed only in one type, and some expressed in the other one. And there will be genes, some that are not expressed in either of the cases here. Okay. So my objective is to know. We don't have to know these things, right? I don't need to know that. I also don't need these. What I would be interested is either typically either what are only specific to B or what are only specific to A. So if I can have a way of cloning just these genes, then I will have a smaller size of library to work. When the library is large, difficult to work. It's tough to work with so many clones that usually it goes to thousands. And imagine if we have to go through all the thousands of clones and then see, then it's going to be difficult. We can also, if it is smaller, then it's easier and less time consuming to work with. So subtractive libraries come in that way, that you can say, for example, I do like this, A minus B. What all genes will come in there? Genes that are A specific, right? I am subtracting B from A. You could also have done B minus A. Then you will get B specific genes. So the one that you are testing is called the tester. Okay, I will try to use it before. So this is a tester. And the B which you are subtracting from the tester is called as the driver. In the second case, in this one, B is the tester and A is the driver. Okay, so subtractive library is based on subtracting one gene set from another cell type, so that you will get only a small A specific or B specific type of genes, and you will generate a library based on that. Only small, very small number of library you will have, and then you can work on those. So I will just take this and then I will repeat this in the next class. Try to see how you can find out. My objective, as I said, tester is this. So for simplicity, I'll put A minus B, so that I will get A specific genes only. I do not want anything else. So tester would have been A is the tester and B is the driver. I'm trying to find the difference between these two, which RNA are produced or transcribed, or I'll put it like this: the transcriptomic difference between A and B is a subtractive library. If you have made a library clones of that, then you'll do something like that. So what they do is they make a cDNA library of driver mRNA or cell type B. All the cDNA generated from it will be having a biotin added to it, and the tester mRNA is also made double-stranded cDNA. The difference between cDNA of B and A here is that B is associated with biotin, and then you mix it up, and then you, upon mixing, you denature and reanneal. Then these two are related species. They are from the same organism. Like I was saying, for cell type A and cell type B, the sequences are similar. There will be differences, of course. Those that are specific here, what you may have is the top strand may find the bottom strand, and it might find my driver link like this. Okay, a double-stranded cDNA, which is composed both derived from cell type B, as in the case. In some cases, when you have genes that are common between cell type A and cell type B, they may form hybrids like this. Okay, hybrid in the sense one is labeled with biotin, the other one is not labeled. And there will be some that do not have which ones are likely to have not have any partners in this one. In B is those that are expressed in A but not expressed in B will remain unique. And also, I forgot to mention, usually the driver mRNA is taken, or the driver cDNA is taken 10 times higher, so that whatever is common, you are trying to, when you take these higher, you are trying to encourage the formation of driver tester hybrids. When will driver tester hybrid form? Only if there is complementarity between the driver and tester mRNAs. Those sequences, such as these, that do not have expression, that do not have any, that are not expressed in cell type B, will remain as unique tester cDNA. And now you have you can pass all these three through the column using streptavidin. So biotin binds to the streptavidin and will retain all the driver combinations, and also the tester driver. They will be retained in the stationary phase, and the through the mobile phase, or the unique testers that do not have any biotin they will pass through the column, and then you can isolate the cDNA and construct a library. By doing this, we have reduced a large number of clones. Library size is drastically reduced. We have removed anything that has not expressed in A, that is not there. Anything that is also expressed in B is also removed. We will only then get just A specific genes. We have subtracted A minus B, and we constructed a library. That is called as subtractive library. Okay, I will take up from here in the next class, subtractive library and race. And I think it's also time for us to discuss about northern blotting, southern blotting, and western blotting. We have an important question associated with that, and we should try to do those in the next class. If you have any questions, I'll try to take up in whatever we have done today. I just go through where I was. I also forgot to mention, I think I told you to find out what are the pros and cons of cDNA library and genomic library. You are supposed to find it out. Please make a question of it and try to find an answer for it. Any questions, please, you may raise, and then I will try to discuss on that.